right, so we are recording. Again, what we talked about last time was the primary active transporter. This is the sodium potassium ATPase. You can see up here sodium potassium. It should say ATPase if you want to be formal about it. It is known as, a lot of times just fondly, it's known as the pump. Okay, so let me just talk about another very interesting characteristic. As we talked about last time with those top hat questions, usually the pump does hyperpolarize the cell on a regular basis. So if you block it with the drug Wabane, we talked about this, it was one of our top hat questions. If you block the pump with Wabane, you will depolarize the cell. You actually do see that in the laboratory. But normally, when it's not blocked, you can see here, I'm going to read from this part, it does contribute to the resting membrane potential, producing hyperpolarization. Again, there is a net movement of one positive charge out of the cell with every turn of the pump, producing hyperpolarization. Now, one really interesting fact as well, this tells you how important the pump actually is. 40% of all ATP that's produced in your cell by mitochondria goes to running this pump. 40%. That's why I call this the engine of every cell. It is responsible for setting up that concentration gradient. Sodium is high on the outside, low on the inside. Potassium tends to be much higher on the inside relative to the outside. And it is the pump that is establishing those gradients. There's a lot of energy in that sodium gradient. And we're going to talk about secondary active transport today as well. Pretty good? Any questions about the pump? Pretty good? All right, so what I'm going to do now is, we've already gone through these top hat questions. Don't worry that before you have your exam, I will open up all the top hat questions for review. You'll be able to see the questions and the right answers again. All right, so let's move on. This is another example of a primary active transporter. This is the F-type ATPase. F actually stands for factor. And it's a very large primary active transporter. This is the F-type ATPase. I'm going to talk about it as an ATPase first. This is under active transport, your lecture on Canvas. You can see that there are many different subunits. There's a large F1 subunit right here. This is responsible for binding and hydrolyzing ATP. Then there's a stalk subunit and a subunit that is transmembrane. It has transmembrane spanning domains. So it's kind of inserted into the cell membrane. And what you see here is these F-type ATPases tend to hydrolyze ATP. Now, it doesn't say that here, but it uses the energy from ATP to move protons against their concentration gradient. And if it's in this direction, then we call it an ATPase. But this is also called an ATP synthase. I find this fascinating. How many of you guys remember this cascade of events? Does it bring back memories of Biology 1009? If you took that, yes. This is the electron transport system, right? And these components of the electron transport system use this is an aerobic pathway. Actually, you can see transports protons into this intermembrane space until you get this very high concentration of acid, of protons, within this space, within the mitochondria. So imagine that this interior part has really high concentration of protons, very, very acidic. Then your mitochondria actually use the high proton concentration, right? Now there's a proton gradient, and it runs this ATPase backwards. So it uses the proton gradient to make ATP. That's pretty cool. I think that's very cool. So this is an example of the F-type ATP synthase. And for every glucose molecule, if you remember that oxidative phosphorylation pathway, for every glucose molecule, this system makes about 34 to 36 ATP molecules. So it makes an enormous amount of ATP molecules. Remember, those are your energy molecules. There's a lot of energy, potential energy, packaged in ATP. All right, so that is an example of an F-type ATPase, or run backwards. It's a synthase. And I want to talk about another one. This is an example of a proton pump in your stomach. These are parietal cells. We're going to get to this when we get to the GI tract. I find this fascinating that, again, this is in your parietal cells. You have the capability, after you eat a meal, to pump protons into your stomach until your stomach compartment reaches a pH of 1 to 2. That is extremely acidic to help digest your food. So not only are you pumping protons by this mechanism into the interior of your stomach cavity, but chloride follows. So let me say that one more time. You're literally transporting hydrochloric acid into your stomach after you eat a meal until your pH is almost 2. So we're going to talk about that when we get to the GI tract and talk about how your stomach protects itself from that very low pH, that very acidic environment. And there are medications that we'll be talking about. You don't have to memorize it now. This is just an example of an F-type primary active transporter. But omeprazole, if you guys go to the store and you look, that's the generic name. I think it's called Nexium or the purple pill. That inhibits this pump for treating what? Acid reflux, right? So we'll talk all about GERD, which is the gastroesophageal reflux disease, when we get to the GI tract. But this is the pump that they're talking about in your parietal cells in the stomach. It's responsible for literally pumping hydrochloric acid into your stomach after you eat a meal. Okay, so some text associated with that. So this is the last primary active transporter that I want to talk about. These are the ABC type ATPases. ABC stands for ATP Binding Cassette. So obviously ATP is required to bind to this particular transporter in order to make it work. It is a large family of transporters, and it transports many molecules. So one example is called MDR, or the multi-drug resistant transporters. So they're responsible for kicking out medications, a lot of times drugs, when it comes to chemotherapy agents, which is unfortunate. So those are called multi-drug resistant transport proteins. They move those drugs out of the cells against their concentration gradient, unfortunately. And sometimes it limits chemotherapy agents and their action. And then also CFTR. I talked about this last time. CFTR was at one point in the evolutionary process. It used to be a primary active transporter. Then it, I hate to use the word, but it devolved. I would say it evolved into a channel. This particular channel is called CFTR. You can see that there. It's the Cystic Fibrosis Transmembrane Conductance Regulator. You don't have to memorize that, but do recognize it. It is an anion channel. So it conducts both chloride and bicarbonate. Both of them are anions, have one negative charge associated with them.
You said conducts chloride and what?
Bicarbonate. HCO3 minus, bicarbonate ions. So almost, you have a lot of cells in your body, and almost all of them have CFTR. It is important in lung function, digestion. It's important in reproduction. So essentially what happens with CFTR, you're going to see, I'm going to say this, you're going to see a lot of misinformation when it comes to CFTR on the web. So I want to be clear as to what happens with cystic fibrosis. Most of the time, 70% of the time, it is a mutation of the phenylalanine at the 508 amino acid mark. It's the delta F 508 mutation. Delta F 508. That's responsible for about 70% of cystic fibrosis cases. What it does is it doesn't allow CFTR to be trafficked to the membrane. It never reaches the membrane. So if you're looking up CFTR and cystic fibrosis and you see a CFTR molecule in the membrane with a big X through it, meaning that it's just not working, that's not right. It just never makes it to the membrane. Okay, so with CFTR, it gets degraded with the delta F508, it gets degraded in the Golgi apparatus and never makes it to the membrane. All right, now I did actually get a hold of my student, my former student, who just had a baby who has cystic fibrosis. She and her husband are really involved with the Cystic Fibrosis Foundation and anything she said to get information out to the public, because she said nobody in her immediate friends and family knows anything about cystic fibrosis. She really wants to put her story out there. So she said it was perfectly fine for me to talk about Spencer. This is Spencer, you guys. He is about 1 year old now, and a lot of these photos are literally 2 weeks ago. So I want to put a face to cystic fibrosis because I really care about this little one. This is Spencer, and when he was a baby, almost a year ago, he was already diagnosed with cystic fibrosis. Most of the time, it is due to a mucus block in their intestines. They have trouble digesting. Almost, even kids that have mild forms of cystic fibrosis usually have intestinal issues. They have to, because their pancreas isn't working properly, they have to take enzymes to help digest their food. And that's when they're first diagnosed. But my former student was also talking about salt in his hair. If you blow this up when you get home, I will put this particular slideshow on your website, and that'll include, the slides. But if you blow this up, you'll actually see a lot of salt in his hair. I will talk about this after you get going on your problem set. So next week, after you have a chance to really dive into cystic fibrosis, I'll talk about why this happens. There's an old wives' tale of folklore that says if you kiss a baby, this is a Swedish folklore, and their forehead is super salty, then they're not going to be on this earth long. I mean, this was a long time ago. Lots of treatments now for cystic fibrosis, but that's the first part of woe is the child, if you read that little adage at the top. So I'll give you a chance to understand why this happens, and we'll talk about it on the back end. He is a normal little kid. I just want to show you a video of him. Here's...
Hey, you take your meds? Can you take your meds?
Those are the enzymes he has to take before he eats.
Why are you being goofy? I don't know either. Here. Take your meds so we can eat.
Oh, he knows. He knows he has to take those meds. And his mom is so good. So good with him. All right, so I better stop this because then we'll be listening to that. Oops.
Hey,
all right.
Take your meds?
All right, yes. Oh, this is him vacuuming too, you guys. This has nothing to do with cystic fibrosis, but he's super cute.
Why do you look so mad? The house is that messy? You do all this work and nobody helps,
Okay.
huh?
Super adorable. Okay. Oh, we'll talk about Lefkowitz in just a second here, too. All right. So anyway, let's get to he is really adorable. I watch all of his progress. This is the vest that he has to use. So this is actually a vest that shakes him pretty violently. He can't eat about an hour before he has this vest on. He's very used to it. You can see he's actually sleeping through it. So he's been doing this his whole entire life. This actually jolts him because it's helping to displace the mucus in his lungs so that it can help him with the mucociliary clearance, trying to move that mucus up to the top of his lungs so he can cough it out. And this was just a couple weeks ago. That's a nebulizer to help open up his airways. My former student was sick, so she's wearing a mask, and he's got the nebulizer on. She's wanting to make sure that he doesn't get any colds, because obviously with these respiratory issues, you don't want to complicate it with like RSV or something. So I just wanted to really put a face to this disease. And as Spencer progresses through the semester too, I'll give you updates. But this is in real time, and he's really a wonderful kid. So now I hope that this helps you through your worksheet as well. So let's move on to secondary active transport. This does not use ATP as harnessing the hydrolysis of ATP to move molecules against their concentration gradient. In this case, secondary active transport uses the energy from the sodium gradient that's actually established by the pump. It uses the energy of the sodium gradient to move molecules, other molecules, against their concentration gradient. So here is the idea. There are two types of secondary active transporters that I want to talk about. If the transporter is using the sodium gradient to move another molecule against its concentration gradient in the same direction, you can see they're both going in the same direction, but sodium is going from high to low, and another molecule like glucose is going from low to high. So X, whatever solute that is, it's moving this solute against its concentration gradient. They're both going in the same direction. These are called co-transporters, or I said that down here, symporters. Now, a counter-transporter is when the two molecules are going in different directions. These are counter-transporters or antiporters. The newer name is co-transporters and counter-transporters. Yeah?
Is it always sodium that's going to be moving?
It's not always sodium, but I'm going to say 90% of the time it's sodium. Yeah. All right. So, now you'll notice that sometimes, just like with the pump too, there are other primary active transporters that are actually coupled to secondary active transporters. In this case, this is a proton pump, an F-type primary active transporter that is using ATP, the energy from the hydrolysis of ATP, to move protons against their concentration gradient, establishing a very high concentration of protons on the extracellular side. Now you've established that proton gradient, and then the secondary active transporter uses that proton gradient to move another molecule like sucrose against its concentration gradient. So it's using the energy that's established by the proton pump. It's using that proton gradient to move another molecule like sucrose against its concentration gradient. And this is interesting. This is just a graph that shows you that as you increase the concentration gradient, you're increasing the energy for the transport of another molecule like sugars, glucose, sucrose, or amino acids. So as you increase the sodium gradient, you increase the transport rate of these other molecules. So that's very interesting. This is just some review. Symporters or co-transporters transport solutes in the same direction. You guys can take a look at this at home too. If you want to take a look at these animations, they're very helpful. They're not that exciting. They just show that they're going either in the same direction or in opposite directions. Now, some of the antiporters that I'm going to be talking about today too is the sodium-calcium exchanger. These are types of antiporters. The sodium proton exchanger, I talked about that at the end of class last period. And the chloride bicarbonate exchanger. It's going to be really important, especially when we get to red blood cells. Also, some important terms just to wrap this up. What is the difference between electroneutral carriers and electrogenic carriers? With an electroneutral carriers, it's either the transport of uncharged particles or the exchange of an equal number of charged particles. So, I'm just going to bring it back home here. Remember our pump that's in our stomach, the parietal cells within our stomach lining? This is a very good example of an electroneutral secondary, this is actually a pump, but this actually shows you that this primary active transporter, because it does use ATP, is transporting the same amount of charge in one direction as the other direction. So it is considered electroneutral, unlike our sodium-potassium ATPase that is electrogenic, because there's one net charge that's moving. So let's go back here. Electroneutral, transport of uncharged molecules or the exchange of an equal number of charged particles. And then electrogenic, there's a net transfer of charge. So this is our last slide for active transport. This is just summing all of what we talked about with passive, facilitated, and active transport. These are some of the most common players that we're going to be talking about. We did talk about ion channels, specifically the voltage-gated sodium channels, which we'll revisit with action potentials. Potassium channels, those leak channels are setting the resting membrane potential. Chloride channels like Cftr and calcium channels. Then we talked about facilitated diffusion, those carriers. One classic example is the sodium glucose transporter. Again, it transports glucose with its concentration gradient into the cell. That's a type of facilitated diffusion. Primary active transporter, here is our sodium-potassium ATPase, some of our F-type, factor-type ATPases, and then there's some calcium ATPases that are important, specifically in the heart. And then our secondary active transporters. This one, sodium glucose exchanger or sodium amino acid. These are a type of a co-transporter. And then our sodium proton exchanger, sodium calcium, and chloride bicarbonate are all counter transporters or antiporters. All right, we're going to see a lot of this now. I'm going to apply this when we get to cell signaling. But first, let's talk about G-protein coupled receptors. A lot of you may have had this already, but I can't make that assumption. So we are going to be going through GS, GI, and GQ. So here we are, we're talking about cell signal transduction pathways, right here. I'm going to just briefly give you an introduction about receptors. Now, you probably, if you've taken cell biology, you'll probably have seen this already. What is a receptor? Receptors are proteins that receive messages from chemicals like ligands. When a ligand binds to its receptor, there's a cascade of events that usually causes some kind of response within the cell. Most messengers are water soluble and bind to receptors at the plasma membrane, so they're hydrophilic. But some receptors, like hormone receptors, estrogen receptors, these are steroids or steroid-like, and they're hydrophobic. They prefer to be in a lipid environment, and they can easily cross the plasma membrane. They don't even, by passive diffusion, now you know what that is, and bind to receptors intracellularly, sometimes intranuclear. They actually can just go right into the nucleus, and usually, a lot of times, they promote protein synthesis. Okay, that takes about 24 hours. All right, so when a hormone or some kind of, I should say it this way, when a hormone is released, whether it is hydrophobic or hydrophilic, vasopressin is a really great example. When we get to the kidney, we'll talk about osmoregulation and how vasopressin fits in. A lot of times these hormones are just dumped into your circulatory system. But why is it not every cell in your body responds to it? Only specific cells respond to it. And that's because those cells that respond are actually expressing the receptor for vasopressin. So this gives it some kind of specificity. This selective expression leads to specificity in the system. So not every cell is going to respond to a ligand or a hormone, like vasopressin. Only those that are specifically expressing the receptor for it. Today we're going to be talking specifically about G-protein coupled receptors. We'll apply this when we actually get to the heart and talk about the sympathetic and parasympathetic nervous system. I'll just briefly talk about that in a minute. You all know that if someone scares you or you start to run, it speeds up your heart, right? That's your fight or flight response. That's a sympathetic autonomic nervous system response. And we're going to talk about why that happens. Why does that speed your heart up? Why does that, actually regulate cardiac pacing? And it's due to these G-protein coupled receptors. That's why we're learning that now. Everything builds upon itself. So I'm going to give you an overview and tell you how G-protein coupled receptors fit into the big picture here. Let me blow this up even more. Here are different types of receptors, ligand gated, if you will, receptors. This orange first messenger is your ligand. It can be a hormone like vasopressin, or it can be acetylcholine. This would be an example of a nicotinic acetylcholine receptor that's expressed in muscle. Now, for this section, you don't need to know these details. I'm just giving you some examples so you can see how I'm going to apply it later. Right now, it's just about the receptor and ligand, the basics. So our ligand binds to our receptor, and in this case, this is an ion channel. It opens up an ion channel, allowing for sodium or potassium or chloride or whatever to move across the membrane. So this is actually called ionotropic. Ionotropic. Ionotropic receptors. These are receptors that are ion channels. These are receptors that are ion channels. So there's many of them in neurons, in dendrites. We'll specifically be talking about the nicotinic acetylcholine receptors when we get to muscle, skeletal muscle. I didn't draw it out because I don't want you to memorize it. We'll get to this when we get to muscle. Nicotinic acetylcholine receptors. All right, so going back, let's talk about some other receptors. Sometimes a receptor is a kinase. A kinase is anything that phosphorylates stuff. Don't you love that? Scientific term stuff. Okay, so when the ligand binds to its receptor, it activates this kinase within the receptor that can phosphorylate proteins, like docking proteins, that then initiate a cascade of events and a global cellular response. Sometimes when the ligand binds to its receptor, it associates with an external, this is intracellularly, but it actually binds to another kinase, this is a JAK kinase, that then phosphorylates other stuff, right? This protein is then phosphorylated, and then again, it leads to a cascade of events and a global cellular response. Today, we're going to focus on G protein coupled receptors. In this case, when the ligand binds to the G protein coupled receptor, it causes these G proteins to change their conformation, activating them, which activates other intramembrane proteins that leads to a cascade of events. Okay, so we're going to do that today. I just want to make sure that you all know that which G proteins we're going to study in this class. You need to know GS. S stands for stimulatory. You also need to know the GI pathway. I stands for inhibitory. And the GQ pathway. And I have no idea what Q stands for. All right, sounds good? So GS and GI, luckily they're almost exactly the same. There's just one piece that's different. You won't have to really memorize anything further when you're studying GS and GI. But GQ actually increases the amount of intracellular calcium levels. There's a global response. Calcium floods into the intracellular space, which leads to a lot of different proteins being phosphorylated and a global response again. All right, so just wanted to show you, this is Dr. Robert Lefkowitz. Oops, let's go to our, this is Robert Lefkowitz. He won the Nobel Prize in 2012 for his studies of G protein coupled receptors. He pretty much characterized and let the world know that these G protein coupled receptors exist, and he characterized many of them. He did win the Nobel Prize for Chemistry in 2012. I actually got to see him when I went to the American Physiological Society's meeting. That's experimental biology. I got to see him one year, and he gave a talk on his latest studies, his research, and you know, it's really, I want to give credit to Dr. Lefkowitz for his role in all of our textbooks now have, you know, this is something that everybody learns in microbiology, biochemistry, physiology, right? It's something ubiquitous that is really important in our understanding of how cells work. So, instead of actually going through GS from these very static slides, this is where I want you to follow along with me. I'm going to talk about these channels, but I like to draw it out because I think it's much more dynamic. All right, so first we're going to concentrate on GS and GI. We'll first concentrate on GS. If this is the plasma membrane, right, and we draw our G protein coupled receptor, our ligand of interest, classic example is norepinephrine and epinephrine. They're mediating sympathetic surge that you feel when you get scared or you have to run. Epinephrine and norepinephrine. This used to be called adrenaline and noradrenaline. So you can see this is what's causing your heart to race. The receptor are the beta adrenergic receptors. They are a typical GS coupled receptor. Again, you don't need to know the details. I just want you to know the pathway, how this works. I'm just giving you some examples so that you won't be surprised when I apply this later on. This receptor has G proteins that are associated with it. We have the beta, the gamma G protein, and the alpha. The alpha is what we're going to concentrate on. Before our ligand binds to its receptor, it has a GDP molecule associated with it. It's like ATP, but with guanine diphosphate. Okay. Now, once our ligand binds to our receptor, there's a conformational change that basically releases GDP and promotes GTP to be attached. Okay, so once the ligand binds, it exchanges the GDP for GTP. Now, this alpha subunit is a GTP ace in itself. It's eventually going to break down GTP. But what it does is now it promotes the dissociation of the alpha molecules from these other molecules and the receptor. So it starts to migrate down the membrane. And this GTP is like a timer you just set with your kitchen timer. So tick tock, eventually the GTP is going to hydrolyze back to GDP. But when it is in this form and GTP is attached, it dissociates, it migrates over, it actually binds to an enzyme called adenylyl cyclase. Adenylyl, YL, cyclase. Adenylyl cyclase. ADENYL. And once our alpha subunit with the GTP attached to it binds to adenylyl cyclase, this enzyme, it's got an ACE at the end, it's an enzyme that converts ATP to cyclic AMP. And cyclic AMP is a really important second messenger. It goes on to activate within the cell protein kinases like protein kinase A. And this kinase goes on to phosphorylate a lot of different molecules within the cell, including Cftr. Okay, so that can actually go and target channels like Cftr and activate it. So I just want you to know too that this is an amplification response. So you get a number of cyclic AMP molecules that are generated, which then go on, each one of those actually go on to activate many more pKa molecules. And again, that's an amplification response. So see how this is just kind of static? But you can see this is the amplification that occurs. Cyclic AMP goes on to activate protein kinase A, which then goes on to activate many different enzymes and many more enzymes. So this is a true amplification global response within cells. All right, so let's talk about GI. Okay, GI. GI, the typical ligand is acetylcholine. And the receptors are called muscarinic receptors in the heart. And they are GI coupled. Pathway is pretty much the same, so I don't have to redraw anything, except when the alpha subunit migrates over, right? So acetylcholine binds to the muscarinic receptors, does the same thing. It's a conformational change. It actually exchanges GDP for GTP, starts the timer ticking, goes over, migrates over to adenylyl cyclase. But in this case, instead of stimulating adenylyl cyclase, it inhibits adenylyl cyclase. So cyclic AMP levels fall below the basal rate, falls, pKa activity falls, and it just shuts everything down. Shuts everything down. So GS stimulates adenylyl cyclase. GI inhibits adenylyl cyclase when it interacts with it. I didn't finish the story here. Eventually the alpha subunit hydrolyzes, right? This is actually a GTPase in itself. It hydrolyzes GTP to GDP, and then once that happens, it migrates back to the beta gamma and its receptor, and it associates with it. So again, it's like a timer. Tick tock, until GTP is hydrolyzed back to GDP. GTP is hydrolyzed, and then it reassociates with the receptor in the beta. You don't need to know the details of the ligand or the receptor names. I will revisit this. You'll see them again when we get to the cardiac pacing unit. But for now, just know how this works. So here's what you need to know. This is the figure. You can see our ligand is the first messenger, the receptor, your G proteins, GDP at rest. Once the ligand binds to its receptor, it exchanges GDP for GTP, has effects on adenylyl cyclase. With GS, it increases cyclic AMP and protein kinase A. GI actually does the opposite. All right, so let's kind of go through this one really quickly. We have a few minutes left, and then we'll be done. Here is our plasma membrane. This is GQ. I'll make it simple. Here's our receptor, our ligand. Good example, I don't even want to write it because it just makes it, I think, more messy. These are Adrenergic receptors. This ligand could be UTP. It's a typical GQ coupled receptor. When the ligand binds to its receptor, just like before, it has a beta, gamma, and an alpha. At rest, it has a GDP attached to it, but once the ligand actually binds to its receptor, it exchanges GDP for GTP. In this case, the alpha subunit then migrates over to an enzyme called phospholipase C. Phospholipase C. It is an enzyme in itself that converts. I just want you all to know that this is PIP2. It's actually 4,5 phosphatidyl inositol bisphosphate. PIP2 is just fine. PIP2. PIP2 is converted to DAG, diacylglycerol, which is membrane bound, and IP3, inositol triphosphate. So PIP2 is broken down by the enzyme PLC to DAG and IP3. IP3 goes intracellularly. Inositol triphosphate. That actually binds to receptors in internal stores. You got lots of calcium in the smooth ER. This is calcium 2+. Binds to its receptors. Calcium floods into the intracellular space. If you use Cftr dye and you're looking at it in the microscope, it just lights it up. These dyes called Cftr just light the whole cell up. It's a global response. And it's actually a positive feedback mechanism. It amplifies the response because calcium also activates IP3 receptors. So the more calcium that enters into the intracellular space, the more calcium's released. It helps with the amplification response. So now, oops, I know we're 1 minute over. Now you can actually follow along with this particular slide. And we'll talk more about this on Monday. Remember, your exam is postponed till October 2nd. Have a wonderful weekend. Yay, it's Friday. See you on Monday. Oh, no worries. No worries, no worries. Thank you, thank you. Oh, Meghna, there were some that were left on that table there. Yeah. Thank you.
I had a question about slide 34.
Okay, yes. Okay, so this is it?
No,
on the other one.
Yeah.
Okay, so it's this one.
Yep.
And this one.
Yes.
Okay, yes.
So is this combining primary and secondary?
Yes, exactly.
Okay.
They're coupled.
Okay.
Yeah,
Are they always coupled?
A lot of times they are.
I
because
thought secondary only uses one protein.
it does. So imagine this though. The secondary active transporter has to use the energy from a gradient.
Right.
And the gradient's established by the primary active transporter.
Okay.
Yeah.
It's not just activated by one, like Na plus going from high to low in that same protein?
It is, but you have to have the gradient
First.
for it to work.
Okay.
So if you block the pump and the gradient runs down, then it's not going to be able to move that molecule against its concentration gradient. You'll lose that energy.
Okay, so it doesn't even produce enough energy?
Yeah.
To help a second molecule. It needs a pump?
It needs the gradient.
But I thought naturally.
Yeah, right?
Yeah.
Not always. Not naturally.
You're going to that East Bank, right? Yeah. I'll ride the bus. Oh, you were?
This gives you an idea. There is a basal activity.
Whether it's like this how susceptible like exam question, I'm just gonna use a couple of those on this one.
GS increases the activity, and GI.
Have you taken biopsy yet? I'm taking biopsy one right now. Oh, you are? Okay.
So
Are you
just
taking it
put it on
through
for
second?
yes.
Okay.
You have a sympathetic response, but if you have a very sympathetic response,
I'm taking it right now, but it's just like the regular one, so I guess I don't know if it's comparable or not. I mean, I'm sure like the material is, but I don't know what brand. Yeah, or like there's like more of a focus on certain
I
things.
was reading before class. University of
We're on.
Michigan
What are we doing?
Med Lab
We're basically just on.
email sent. I broke down the email to Nelson. So Michael Stratman is the person who named the GQ protein.
Oh, cool.
Instead,
Oh, that's awesome.
he couldn't use the front end of the alphabet because the O letters were so
Right.
quick. Subunits. And GZ seemed, or had been taken, and X seemed too obvious, so we went with Q.
Nice. Can
So it's
you?
more of a, yeah.
Oh my gosh, I am. You know, I have been teaching this class for 12 years.
It was just someone's preference. They're
That's
like, okay,
awesome.
yeah, it makes
Can you
sense.
send me that exact
will,
quote?
yes.
I am going to say that to the class
and
on
then-
Monday.
So
Was there a third one?